Okay, so yeah, I'm trying to explain potentially everything that comes afterwards about the hardware and the software to automate stuff and how to build stuff. And we also have a session in the afternoon to teach you how to use these elements to build a fluorescent microscope and also how to automate it. So I'm here from two different perspectives. So I think I should maybe talk about the conflicts of interest, but everything is open source. So we have a little company around it, but I'm still also a group leader at the Leibniz Institute of Photonic Technologies in Jena. And yeah, I'm just diving right in, so I don't have time. So tools, I think everybody knows some of these, like ChatGPT, Google Colab, I don't know, like these tools make your life easier when you do software programming. But the same is also happening on hardware side. So over the last, I don't know, 10 years or so, when like 3D printers became like a thing, there's not only printers, but like you can print your PCBs, you can mill, you can order stuff online, and you can also do injection molding nowadays with desktop stuff. And this is, I think, the base for everything that's possible now also on the hardware side. I mean, software is getting cheaper and cheaper, but also hardware, it becomes available. And it has started probably with this little thing where we essentially just tried doing super resolution with a cell phone. And we kind of stressed a bit like what is like a phone can do. In that case, it was a monochrome camera, so you get more photons, you get better signal to noise. And we basically replaced this 20,000 euro camera. It's an EMCCD with just, well, monochrome camera from phone. And we already got like 80 nanometer. That was everything cool. We also did some neural network processing, but still you have like the whole body of your microscope, expensive, bulky. And so what we try to do is basically try to convert this whole thing into something that's more affordable, available, and accessible. So that more people have access to the tools you need for biology to discover everything that Juliana just has mentioned. Because microscopy, it's in a way a bit bizarre because it's very, I mean, the components are not really available. You can get them from, I don't know, Amazon, eBay, AliExpress and stuff, but it's very hard to put them together. And Thorlabs and Edmund Optics, they don't really help you that much. And so in the end, if you, well, this is a rendering, but I mean, you have seen optical tables, the situation can get that far very easily. So, and we are about to help. And so we got inspired by this little guy here. So the Arduino, probably who has ever seen this? Yeah, okay, so the majority. So I'm coming from the background of electrical engineering, or I did my apprenticeship as an electrician. And so doing just like a very simple thing to get a LED blinking takes you a very long time. You have to develop the circuit board, print it, and then eventually also put the components on. Here, you just plug it in, the USB, it works. And the cool thing is open source. You have a huge code base from open source communities, which you can just use for your purpose. And so this is something we wanted to translate onto optics. And luckily, so if you observe a common microscope, you have components like illumination, samples, objective lenses, and also camera. And they form this modular framework already. And so the logical next step was basically just drawing boxes around it. We called it UC2, published that right in the Corona pandemic where everybody was stuck at home with printers. So everybody was printing cubes. And then now we have this kind of open source community where people actually also contribute to the system with new setups. And that's pretty cool. So it's like a library of parts, similar to like a Python library where you can just import your modules, put them together to build a fluorescent microscope, for example. And the cool, well, to me, cool thing is you can basically place anything in these cubes. It's really just a skeleton, so there's no fanciness around it. And so it can be, for example, an LED array, it can be a laser, it can be an objective lens. And these cubes basically just make them compatible with each other. So you have little puzzle pieces to put them in layers, then you stack them, and then you can build basically any microscope you want. And so you start with a cube. We help you get starting with some wikis and some tutorials. I think for everybody who's there in the afternoon, you can see how easy that can get. We also have some boxes. Either you print them on your own or you buy them through the startup. And then this whole ecosystem is growing. So we started with geometrical optics for schools only. Now we have also smart microscopy stuff where you have automation. We even have quantum boxes now. And this whole thing is ending somewhere in like a semi-professional way. So this device is also here somewhere, which I have to recreate today. It's a frame where you put your cubes, which you have learned on assembling first, and then into a framework that's very robust and can be used in like a professional setup. And the whole software around it is, well, modular as well, which is, I think, very important. So this is the library of parts. You can find all of them on GitHub somewhere. If you don't find that, there's a forum where people can help you. There's also now communities actively developing new tools. For example, some people in Chile, and they cannot just get stuff from Thorlabs as we would do that the other day. So they have to print the parts in order to do their science. And actually that's pretty cool. Same for people in Nigeria. And like all across the world where people are doing these workshops. And I think something very special is here happening in Madagascar sometimes. So they have workshops with 2,000 participants, and then they teach how to do microscopy in these classrooms, which is pretty cool. So last year we did, or like 2 years ago, did a workshop in Nigeria. And so these parts, they are now floating all like somewhere in Nigeria. And once in a while we see somebody teaching microscopy in the middle of it's close to the Sahara, so desert, and even in French, which was very, very nice. And so there's also people taking this up. So Seed Studio is a big electronics manufacturer. So they kind of took the idea, since it's open source, and they manufacture now this microscope here. So basically that's our mission: make microscopy somewhat available. And maybe for stuff, so people who use more sophisticated setups, so Lightsheet, for example, who has known this technique before? Again, half the class. So this is basically a method for doing 3D microscopy. So instead of illuminating your full sample all at once, you slice it by illuminating your volumetric object from the side with a plane, like a light sheet. And then while moving the sample through the light sheet, you can capture plane by plane. And then afterwards, you basically just stitch your volume into 3D space. And with the cubes, it's becoming really, really simple because you have just a laser. In that case, it's a fiber coupled laser, which is then being shaped into a line, like similar to the cashier, and then you illuminate your sample from the side, and then from vertical, perpendicular side, you have your infinity microscope, and it's really just putting blocks together. What you see here on the right-hand side, that was a zebrafish, where you then do the slicing with automated stage. You do the imaging, and then afterwards you can do the stitching using open source software. And then we had this collaboration with Manu Prakash from Stanford. We wanted to do that for marine organisms. And there the challenge was to continuously measure 3D samples, like not only because like usually you mount your sample in agarose and then that's it basically. And so we kind of, very easily modified the setup by having a tube that is constantly rotating and a pump that is creating a flow. And so what's happening, the sample is spiraling through the sample, like through the focus. And then you can continuously measure, and then afterwards, like deskew and stitch the stuff in 3D again. It's not quite there yet. So this is like an algae. So you see, so we try to invert the hologram here. So this is kind of an inline hologram. And then from there, from the fluorescence is rather easy. It's just tomography. And so that's also why it's called Holy Sheet, for the holography and the light sheet part. And then eventually you could get label-free information from your sample. So whoever is keen on taking up the computational stuff afterwards, that would be very nice. We can also do Storm. Storm is this thing where you have individual fluorophores blinking randomly over time, and then you can basically draw the locations, and then you get very high resolutions. So that was a group in Berlin. So they took our microscope idea and then built a microscope around it. And so we have this software that's called ImSwitch that's also open source, developed from the group from Ilaria Testa in Stockholm. And so we basically took over after the person has left. And so we tweaked it more and more. So this is also compatible with Micromanager or PyMM Core Plus, and you can add all kinds of different components. And for us, it was important to also have an open source controlling mechanism that is easily accessible. And so what we did is we have an open source firmware where you can actually even use that by just using your browser. So you don't need to install anything. You only need parts that you can buy from Amazon or eBay or some of your beloved suppliers. You plug it in, you flash the firmware through your browser, and then you can control that through the browser. And so we kind of had a look, okay, what are the most important parts you would need for a microscope? That's usually a stepper motor for moving the stage in XYZ, maybe some pumps or something. You have LED matrices for doing, for example, quantitative imaging, lasers, galvos nowadays as well. So you can build also a small confocal microscope with this. And then from the control side, it can be controlled via Wi-Fi, USB, or also like dedicated drivers from Python are available. And so this is all baked into one single firmware, and then you can choose which module should be activated. And then once you have all the ingredients together, well, you've learned what's a cube, you've learned how to put stuff into the cube, and then you learn how to combine these different cubes. Then you can put this whole knowledge into the frame, and the frame is essentially just something that is holding the cubes and then your sample, so that everything becomes a bit more robust, let's say. And so this is also open source. It's not fully done yet, so it's still in the development phase, but it's all based on the same idea about modularity and the same firmware. So and why is this cool? I think you can, for example, just develop like a single layer, similar to like and then I mean, my vision or like my dream would be, okay, there are some people having a very cool publication, and then to make it somewhat more reproducible, they maybe develop that within this framework, and then once you publish that, like similar to Python file that you have accessible during your publication, you have your hardware as well, so that people can actually reproduce the results. And I think we're not fully there yet, but we've started, for example, using Flim, or like others have used Flim. So Flim Labs, that's yet another company. They have modified our layer. And so this layer is a little confocal microscope with their Flim. So Flim stands for fluorescent lifetime imaging. And so then you have just one more modality to get starting with. So it's really just their expertise put into our framework, and then you can just get starting. Something we will learn in the afternoon, I think. So these little microcontrollers, as I said, you can use any ESP32 with any board you want to adapt. For example, with your steppers, you flash the firmware online. So you can go to uc2.io. So everything there is also open source. And then you can control that in the same browser, which nowadays looks even a bit more fancier. But for more sophisticated stuff, so what Suliana was talking about, like if you have an image, you want to compute an action and then have the action on the hardware, then you would need to have some kind of complicated or more complicated framework. And for this, again, we use this IMSwitch. Again, IMSwitch is developed by other people. We have taken it up. And since IMSwitch was or is still implemented in Python, I mean, everybody knows how easy or difficult it may become to install Python on a new computer. So we have decided to put everything in a Docker container. So you basically just download the Docker, you start it, and then it's working out of the box. The cool thing, which we have figured out, is that you can even install the drivers inside the Docker container, so that you really just like download this whole Docker thing as a zip file. You kind of do a double click and it works out of the box from it. And then you can control your microscope, since we exposed that to the network, basically from anywhere. And since it's rather resource less intense, you can also run that, for example, on the Raspberry Pi, and then you can have multiple of these instances, and then you can have multiple of these microscopes because they are rather cheap, and then you can just have a farm of microscopes. That's a bit where we're heading towards. And that's all also part of the afternoon program if you're keen on looking that up. Yeah, and also one mission from our side is like to make not only the hardware simple, but also the software. So we're teaming up with the people from Manu's lab. So Ethan, he and the people from the Planktoscope, they have this mechanism to kind of create an operating system based on your needs. And this is very technical, but long story short, now what we have created is whoever has worked with Raspberry Pi, you have basically just one image file, you put that on an SD card and everything is already pre-installed, which makes like now I think the hardware also a little bit more reproducible, which was, well, always lacking a little bit. So this is basically, so everything like in Docker, Jupyter Notebook. So we're trying to make the hardware tools a bit more user-friendly in the end, I think. So this is just something I wanted to mention because, but I'm running out of time a little bit. So we have worked on some smart microscopy workflows where we wanted to, for example, have an OpenTron, so like that's a pipetting robot that's also kind of open source, our microscope and other software somewhat being orchestrated all together. There's this tool developed from Johannes Roos. It's called Architect. And this is basically a service that is connecting all the different pieces in a distributed network fashion. So if you, for example, have a node with a pipetting robot, a node with a microscope, or a node with some image processing, where I just take Elastic because it has a nice logo. It can talk to each other no matter where it is. So for example, our cluster is somewhere in Berlin. This does the heavy image computation. Our microscope is in Jena, and the OpenTrons is also in Jena, but then all the data flows managed magically by architect, and then you can just do the heavy computation not on the Raspberry Pi because it's not so computation equipped somewhere else, which is quite nice. And this slide I just show because this may be the translation to Katrina's work later. So we have teamed up with Wei and their colleagues on this hackathon last year in Madrid. And so this is just something to show why open source is pretty cool. So we basically took another software that was open source, combined that with our open source software, and what you see there is like you chat with the microscope or with kind of the sample. So I asked it to come up with a algorithm to do autofocusing, and since this ChatGPT instance knew all the functionalities like move the stage, take an image, and also come up with a way of measuring the contrast, it came up with an algorithm that was then executed on the microscope and then brought the sample into focus. I think it's a less relevant experiment, but you can do all kinds of things now with, for example, the Vision API to really ask, okay, what kind of cell do I see? So just some abilities, or like here in that case, there are some people, they are tracking roots to see whether there are nematodes. So these are like little parasites. And this is one part where we help out with smart microscopy. Okay, so I'm just giving that. So the afternoon is about this setup here. So we have a bunch of boxes, which has parts in terms of software, hardware, optics, everything you need to build this microscope here. So this is a fluorescent microscope. It's relatively simple. And hopefully by the end of the day, you know how to not only build it, but also maybe to modify that. So for example, if you have already parts in your labs drawer, how to design your CAD files in order to print some adapters. Yeah, and this is it. So this is our team in Jena. Actually, it has grown already since then. And